today. First, I would like to welcome Thomas Norman from Memorial Sloan Kettering Cancer Center in New York. He's done some really amazingly influential work on genetic perturbations and interactions. And so today we're going to be hearing about where are the perturbation phenotypes. So thank you.
All right. Thanks, Ben, and thanks for inviting me. So I took the assignment rather seriously. So this is not a research talk. This is more of an open question, which means that I do not know the answer to the question, and I'd be delighted to talk to people who think that they do know the answer. And I think it is a question that lends itself to sort of glib and simple explanations, but then when you are pressed to actually explain what you mean, it is not easy to come up with an actual way of doing it. And so it's called Where are the Perturbation Phenotypes? To introduce myself, actually, it's fun to be back at a sort of math environment. I myself am a lapsed mathematician, but am now essentially a technologist. And the thing that I'm best known for is developing this perturb-seq approach, which I did during my postdoc in Jonathan Weissman's lab. Ben gave a nice overview of this yesterday, but basically this is a CRISPR screen where the readout is single-cell RNA sequencing. It works like any other CRISPR screen. You take a library of CRISPR perturbations, so we can knock genes down, we can turn them on, and you deliver them by transduction such that each cell receives usually at most one perturbation. And so we're using each cell as a little reaction vessel to test a genetic hypothesis. And then we can read all of those out in parallel by single-cell RNA sequencing. This has this sort of great benefit of causal interpretability where you know what gene you're perturbing and what you're doing to it, and then you can read out its effects in the transcriptome. And so in preparing for this talk, I dredged up this old slide from during my postdoc, when we were first sort of justifying this approach, which is why do this sort of complicated single-cell RNA-seq readout? And so the classic way that you do a CRISPR screen is by fitness, which is an extremely simple and parallelizable readout. But the sort of challenge is, we all know that cell state is this much more complex beast. So I'm sort of visualizing it here as this abstract manifold. And when we do a fitness screen, we're taking that complexity and we're compressing it down onto a line so that things that may be very different in sort of this higher dimensional state space end up very close to each other. And so we're throwing out a lot of information. The hope would be that the transcriptome captures more of that state information. And so it's also biologically interpretable because we know about differential gene expression and things like that. And then taking a step back further still, it's also just a great way of attaching a vector phenotype to each genetic perturbation, and this has generated a whole lot of excitement about machine learning because it gives you something to hold on to that scalar phenotypes do not. And so this is what has motivated all of this work on virtual cell models, which again, Ben touched on briefly yesterday. One reason we might want to do this is the reason that he put forward, which is to understand diseases with complex genetics, because you can't do sort of many combinatorial perturbations at once. The other thing that if we could sort of rationally do arithmetic on perturbations, we could sort of nominate strategies for guiding cells from one state to another without having to do every experiment along the way. And so the primary challenge with these virtual cell models, they've gone through sort of like two hype cycles. First, where they were all released, and then it was like the idea that we're modeling all this data. And then the second hype cycle has been the models don't work, and they're defeated by simple baselines and things like that. And so I think there's sort of lots of work to be done on improving the modeling. And then, but I'm going to stick to a simpler question here, which is ultimately the sort of real fundamental limitation that we have is that these models can only be as good as the data that they are built on. And in particular, I think to sort of not bury the lead, the problem is if we don't have phenotypes for some perturbations, then we can't model them. There's nothing for the model to hold on to. And so this leads to this very simple question, which is what I motivated this talk with, which is how many perturbations actually have a transcriptional phenotype? And so this only became possible to ask in this sort of current generation of experiments because we're now doing unbiased perturb-seq experiments where we target every gene in the genome. Yep.
A question on the metric. So what does it mean for a phenotype? Like 5 genes different?
Well, I'll show that in a second. Or we can debate. That's actually part of the point. So
also define the ensemble of what you mean by perturbations. Like I'm not sure. Like,
yeah. So sure. So typically people are talking usually about knockout or knockdown phenotypes in classical. Yeah. Targeting one gene at a time. Obviously, there are more complicated things, but that's what's being scaled at the moment. So this is the first experiment, the first genome-wide experiment that we did in 2022. And so this is where we knock down every expressed transcript in K562 cells, at 10,000 transcripts or so. And if I were giving a biology talk, this is the slide that I would sort of emphasize. It's like, we get all of these phenotypes, things are clustering according to known biological processes. You can assign gene function, et cetera, et cetera. So like this is the sort of pitch for why perturb-seq is great because it's sort of sensitive to all these different biological phenotypes. And if you sort of go and look at the labels here, you really are getting a great diversity of things. Now, in order to get this, it actually takes a lot of work. And the primary challenge that you're facing is the sort of technical factors. And so one challenge that I think is underappreciated is that even in sort of 4th generation CRISPR libraries, a lot of guides don't work. And so this is the sort of thing that you're always fighting against because then you get sort of mislabeled data effectively. And so we used in this paper this dual guide design where every element in the library is actually 2 guides. And so then you're getting 2 shots on goal. This is to make sure that if one of them isn't working, the other one can compensate. And then we did some further optimization of CRISPR-I. And so with this, the other nice thing about perturb-seq is that you can see that whether your perturbations are successful or not. So we can...
Bulls work.
What?
What happens when bulls work?
You do get a modest increase in total knockdown, but it's not nothing to write home about, I would say. So you can see, and then we can see that like in K562s, which had a sort of 1st generation version of this, we get 85% median knockdown. In the 2nd generation version in RP1 cells, we get 90% median knockdown. So the reason that I'm saying this is that we have a reason to believe that the perturbations are working. We are knocking down the targets to an appreciable level. So then we get to the question of how many actually have a transcriptional phenotype. And so this is where it starts to get a little bit murky. And so I'm actually showing several different measures of phenotype here. And so one thing, okay, the laser pointer doesn't work. Okay, the one thing, so this is the growth phenotype that we have for all of these guides. So you can see some of them these are essential genes. They have a depletion phenotype. That's why it's negative. This is the transcriptional phenotype. So we can see that there's also an association between having a growth defect and a transcriptional phenotype. That makes sense. Essential genes have strong effects all over the place. And we can see that these things are not completely concordant with each other. These are things that have strong fitness phenotypes, but no transcriptional phenotypes. That's because they're dropping out of the experiment. And then these are things that have sort of no fitness phenotype, but do have an effect on the transcriptome. But getting to Yogesh's question, the thing is, if you sort of like then assign whether a sort of binary label of like whether something is perturbed or not, I mean, the sort of simple, what you can do it in 2 ways. We can do it by differential gene expression. So here is that at different sort of thresholds. And then this purple actually is another way of doing it. This is using a test on energy distance in PCA space. So this is a distributional test on the entire transcriptional phenotype. So an attempt to be sort of very general about what constitutes being perturbed.
It's one particular defined medium solution.
Yes.
So you don't know whether it could be affected in the different,
Yes, yeah, which we'll get to. So that's the question. Yeah.
Can I just understand? So for the ones where you have a dropout, right now it's plotted as if the number of DGs is 1.
Yeah.
But that dot does not exist, basically.
Yeah. That's just so that everything is represented. But so the point is that this, which is that really, if you sort of ask about sort of what has a phenotype, a couple thousand things have sort of truly sort of definitive phenotypes, and then you get the sort of weaker thing where maybe about half of them at most have something sort of detectable. And then this is actually, frankly, being generous, because if we want to do this modeling thing, these sort of very weak transcriptional phenotypes are not really much to hold on to. Maybe they are, but it seems to raise questions. Okay, so this is like the first experiment, maybe teething pains, new technology, etc. I'll show you another experiment from my lab more recently. This is where we did something a completely different, sort of opposite direction. Here instead of knocking genes down, we activated them, and we activated all human transcription factors. And the idea here is that transcription factors, surely those will change the transcriptome. And so again, we face this sort of technical burden. So if CRISPR-I is a pain, CRISPR-A is a pain in the ass, the the guides here really don't work. And so we have to use this brute force strategy where we're targeting 1,836 transcription factors. We target them 6 times each in this library. So it's an 11,000 guide library. And you need that because many of them are just truly inactive. So if half this library does nothing, which is just money that we're flushing down the toilet. Nevertheless, we can activate about 1,300 transcription factors within a sort of physiological expression range using this approach. In fact, I think our success is pretty complete because we can sort of look at what chromatin features identify things that are susceptible to CRISPR-A, and the things that are missing here have sort of simple explanations. They're like methylated promoters and things like that. Okay, so we can activate all of these transcription factors. And then again, if I were giving a biology talk, this is the slide that I would focus on, where I emphasize the successes. This was in fibroblasts. So we can manipulate these transcription factors and cause these fibroblasts to go into these states that they exhibit in vivo. What?
Previous plot, can I quickly understand this?
Yep.
So every blue line, this is every data. You haven't done any quality control on which ones got activated or not.
This is transcription factors that are activated basically from nothing. So what we're doing is comparing the induction level by CRISPR-A to the endogenous expression range. And what you see is that it pushes you towards the top end, but not over. And the point is that we're not over here. I'll comment on this in a moment, where sort of ORF overexpression puts you. So we can activate transcription factors and cause state changes. It's all very neat. But again, I now want to sort of linger on the technical details. So we did this actually in 2 cell types, RPE1s and fibroblasts. We can activate, again, about 1,300 transcription factors. In each cell type, only about 300 of these transcription factors have strong effects on the transcriptome. And it's very cell type specific. And so the overlap's only about 137. So extreme context dependence. And for people who then will say, we're using CRISPR-A, it's kind of too subtle. If you look, there's the paper from the Broad using the Morph library in HESCs. And so that's overexpressing things using ORFs with an EF1 alpha promoter, so like very high expression. And then their estimate is 496 transcription factors with strong transcriptional effects. So there's this extreme gating on producing a phenotype. If you do CRISPR-I, you get more hits, and they're actually also non-overlapping, et cetera. So we have this sort of issue that, it seems that when we do these perturbations in an unbiased way, it's sort of unclear which ones actually do something. And so this can be extended. This is a nice paper. There's been a couple of these. This is a nice paper from Chris Sanders' lab where they just did this sort of energy distance-based test on many perturbations across many large perturbation experiments. And you see across the board, there's this kind of lower mode of guides. So this is the strength of the phenotype, and you see these lower modes of perturbations that aren't doing anything detectable. And so it's not just our experiments. And then I think we can sort of go to the most comprehensive functional genomics experiment in existence. We can go to the DETMAP and ask how many genes are they getting phenotypes for. And so there's sort of two ways that you could look at it. If you just look at what things show sort of strong conditional essentiality somewhere, that's about 5,000. Genes. And then there's this was a nice effort to try to sort of correct all of this data for noise and confounding. And they sort of identify phenotypes for maybe about 10,000 genes, sort of producing this embedding over here. Though I will say that this is probably an optimistic number, because if you look at this embedding carefully, you'll see that there are things that don't make biological sense. So yep.
How long do you typically wait from perturbation to the readout?
Yes.
Phenotype, it takes days, but.
So this is one of the problems, and this is one of the things that so I'm happy to now, so I've made my point. Many of these things don't have perturbations. The remainder of the talk is just sort of candidate explanations, but this is one of them. Obviously, different things have a different time scale. By nature, the longer you run these experiments, unfortunately, the strong perturbations will deplete. And so you get the sort of artifactual thing. So usually for CRISPR-I, people, I think, usually run in the range of a week. And then for CRISPR-A, it's a bit longer because you have to wait for things to turn on and stuff like that. So one possible explanation is just that we're waiting not long enough to catch some of these things.
Tom, can I also add a point to your previous plot? We don't know the explanation, but when you do the same perturbation across two completely different cell types, and you just present your perturbed response in latent space and use a classifier or some off the shelf, it doesn't do too bad on out of distribution prediction. So the same perturbation in two completely different cell types, it can recover the perturbation with pretty high, with similar F1 score as within perturbation. So even if from the metric that you showed that the pie chart or the Venn diagram is not very super high overlapping, there are potentially some latent features. I don't know what it is, but
I agree. I think it would be interesting to know that. I would say that by the traditional metrics that people use to identify things, so that would say that the metrics are wrong.
Yeah, so for example, like one thing we notice is in most of these log fold change analysis, we are very interested in like 10 genes which are very high log fold change, but there could be 100 genes which are super low log fold change.
But when the claim that you're making, are you truly, is your universe all perturbations? Because I could tell you many things work well on highly expressed genes and common essentials.
Like we haven't done a full systematic analysis from 6,000 perturbations, which is covering a lot of perturb-seq data sets.
Yeah, but I'm telling you about 5,000 have strongly selective.
Possible, right?
So I think the question is, can we expand outside of essential gene or things that are adjacent to being essential genes? So I don't have an answer to this. There's a bunch of different explanations. I could just go through a couple of them in the time. I don't know how much time I have left. So the one thing that I think will jump to mind for people, it's all technical. Single cell RNA-seq is noisy. We don't measure very many cells per perturbation, etc. This, I think there's emerging evidence that it's not the case. So one example is Alex Marson's lab put out this primary T-cell perturb-seq experiment. This used 10x flex, which is a much more sensitive technology and also distinct. It measured millions of cells per condition, so about 575 cells per perturbation. It had 3 different conditions. This is their estimates of perturbations that are active, and in fact, it's lower than in these K562 cells that I presented earlier. And also the fact that and I'm aware of benchmarking efforts from various large initiatives that I think point to similar findings. So I think the other thing that's been raised, maybe it's all growth, it's all in vitro conditions. So we grow cells in sort of very permissive contexts, and maybe that's sort of suppressing these phenotypes. There's a well-known literature in yeast that if you just take yeast and put them in different environmental conditions, it will elicit a phenotype for every gene in the genome, essentially. I will say it's unclear that that's true for mammalian cells, which are obviously supposed to be part of a multicellular organism. And I will, in a bit of a shots fired moment, I will sort of mention there's a big push to doing in vivo perturb-seq now, with the idea that, oh, maybe that's where we'll get everything in context. And my initial take on that would actually be mildly pessimistic. And I don't want to, this is a beautiful paper. They just happen to have a very useful figure for my argument, which is, this was an in vivo brain perturb-seq experiment where they're knocking down genes associated with this 22q11.2 deletion syndrome. And what you see are this is the differentially expressed genes for their different perturbations, and you see these 4 outliers. These 4 outliers are all common essentials. So again, we're sort of getting these common essential genes. Okay, so I'll just, there's also compensation as a possibility, and it's possibility that the transcriptome is not an adequate readout. I think I'll just rest at the end with the thing that I think is the interesting point here, which I'm sure many people here are interested in as well, because I think this is where this field is. So the basic problem is that there's lots of these different explanations that you can come up with. You can come up with technical improvements, you can come up with changing the environment, combinatorial perturbations, different readouts. This itself is a sort of combinatorial enterprise of different possible experiments. And right now, we don't know how to rationally deploy these. It's clearly not possible to explore them all systematically. And so as we sort of try to get these diverse data sets that we're going to need for these sort of very large perturbation modeling things, we need to figure out a strategy. So if anyone has ideas about how to do this kind of experimental design, I would love to talk to you and to test them. Yes.
If you were to do like a new screen, would you want to do some kind of combinatorics, which you have done some before? But what is the most information-heavy next screen you?
Yeah. So of course we're and others are pursuing combinatorial experiments. I think the funny thing is there's been again some leading indicators that the naive combinatorial strategies don't work. So I think I don't want to. Pick on people. There was a large genetic interaction map published by the University of Toronto group that does all of the big GI maps. And they use this design where they multiplex essentials with the whole genome. And I think that actually ended up having less signal than you'd hope for. Yeah.
New questions, but we'll have Jessica come set up.
Yeah.
Why are you basically choosing boring cell types doing a transcriptional readout as opposed to more interesting cell types or things for which the cells are screened to resist a challenge or not to?
Right. So I will take issue with describing fibroblasts, which are dear to my heart, as boring. But so I do think we have in my lab we do stuff in primary cells, to be clear. But obviously people screen in cancer cell lines because they're what's tractable to do at scale. But I think of course the sort of drug or environment challenges are ideas. I actually have also heard that drug-based genetic interaction screening has been sort of less informative than hoped. I think the problem is that each of these, it's more like an experimental design problem, right? How do you come up with a condition where you know a priori, or at least have an indication a priori that you're going to elicit an interesting phenotype? And I don't think we have a way of doing that right now.
I just wondered if, sorry that I missed the first part.
Yeah, sure.
It seems like the thing that you'd want to do is then take all of the information you have about what hits essentials and avoid those, so that you have a chance of actually getting something else. So like if I were doing a combinatorial, the last thing I would do is take the things that I think will hit essentials. I would get rid of those and just try to focus on the rest.
Yeah, well, then you run then you run into the challenge that like the naive expectation is that interactions are rare, right? So like you're doing a huge number of measurements. Yeah.
Combinatorics.
Yeah.
just if I were going to kind of try to increase my sensitivity towards things that are not the essentials, is there a way to, like, does everything hit an essential in some screen, or could you just avoid the ones that tend
Yeah,
to
I think that's kind of an open question. That's, like the both actually, there's this sort of Pritchard's omnigenic model that sort of everything filters through these core genes. And then I think Ben actually yesterday had an interesting thing about these soft modes, which is maybe a different take on the same idea. So yeah, I don't know. Maybe a lot of it does filter ultimately through effects on essential genes, right? In which case, then the essential genes are the guideposts that we want to some extent. Yes.
How does this finding affect bulk RNA sequencing results, where whenever you do anything, you look at the cells,
Yeah,
and you get hundreds of differential?
yeah, it's an interesting point, and that is where I will allude to sort of technical experiments that aim at scaling this up, and I think have not found the sort of magic. There is also the thing I've always wondered actually if bulk RNA-seq itself has sort of biases in what gets returned as hits. If you do bulk RNA-seq across like 1,000 different perturbations, do you get like the same gene signature across a lot of them? I mean, this is one of the things that people have seen with these perturbation prediction models, that if you predict the mean of perturbations, period, you do pretty well. And it seems that cells have a sort of set of genes that like to respond to almost any deviation from homeostasis. Yeah. Hey, Brian.
Hey. So I wonder if you know any of these same numbers, but in post-mitotic cells? Because I think that...
Yeah. No, I do not. We did an experiment in contact inhibited cells because I was like, oh, it's just that we need to not have them under active growth and division because it's this very powerful normalizing force. We didn't get a big difference, but it wasn't a super systematic effort as well. So yeah, I don't know. I think the challenge there is that then degradation itself becomes very variable, and I think actually doing CRISPR-I is hard because you'll be waiting different periods for things to deplete. But yeah, I think it's a cool direction, and it probably makes more. Although the in vivo result that I showed, obviously that's post-mitotic. Okay, anyway, I will get off the stage.